Okay, I am super excited to have James here today to talk to us about his work. And James and I have been collaborating a little bit on the rock pools enrichment and hope to do more. So James did his undergraduate at Eckerd College in Florida, and then went on and did his master's and PhD at the University of Florida. Did postdoc work at Boston University and Washington University in St. Louis before starting at VCU in 2007. And he's now the Assistant Director and Director of Undergraduate Studies for the Center of Environmental Studies there at VCU. And one thing I think is very exciting is they recently got, so James and collaborators have one of these NSF RCN UBE grants, which is furthering STEM education and research, right? Like the interplay.
Stream and river ecology, and so I have my eye on some of you. Hope we get to talk about that a little bit later. I won't talk about it here so much. But
it's the River Field Studies Network connecting rivers, people, and science through immersive field-based education. And I feel like the work that we do in the department, we're so interested in having students involved in research, right? And I feel like that a lot of the work James does plays really well with that. So super excited to hear what he has to tell us today.
well, thank you, Karen, for that introduction, and thank you all for inviting me. Actually, this is my second time. I was invited here by Chris in 2007. Oh, okay. Yeah. So you guys have been really busy. You have a brand new building, which at least you can keep up with your progress. But and I actually had a really great lunch with the graduate students. I will give some props to them. Fascinating research. They gave me their spiels and fascinating stuff. I suspect they might be even more excited about the research they're doing than you are. Really came across, and so that's a sign of a really, for me, that's a really sign of a healthy program. If the students are excited, then you must be doing something right. So, so I also thank the graduate students for a wonderful lunch. And I titled this today. This is the first time I've actually talked about the rock pools. In this kind of a setting, and I'm really trying to feature student-driven investigation because I understand there might be some students here today that are barcoding copepods. Is there anybody, show of hands, who's actually? All right, so there are a few of you, right? And this whole, everything I'm going to talk about today has its origin in classwork. Essentially, it started as a pure course event research project that then provided foundational data to support NSF work, and the interplay between research and classroom stuff just kind of went back and forth and various iterations. And so really what I'm going to talk about today is very student driven, and we have students to thank for it. And oops, there we go. And so these are sort of the latest cast who I want to acknowledge. These are the current Vonesh lab members. Andy and Josh and Rylan are PhD students in the lab. Brian, Phoebe, Grace, and Megan undergrads in the lab, and they've been instrumental in collecting the data that I'll talk about today in the last couple of years. And but in addition, I think probably one of the best parts of having a little bit of federal funding is that creates a gateway to play with other people. Because if you have a grant, then you can get ROAs, Research Opportunity Awards, to collaborate with really bright colleagues of yours that are at predominantly teaching schools. And so the main, Mike and Jeremy from Radford and I are the putative, PIs on the grant, but we've had amazing collaborations with Brian Bird, who's a public health person at Western Carolina, Kristen Grayson, ecologist at University of Richmond, also spatial landscape person, Todd Lookingbill, and of course the newest person to us here. I'm going to talk a bit about sort of freshwater riverine systems today. And so the bigger, broader context here is that freshwater systems are vital to our cultures and to our society's well-being, but they're darn complex, like most ecological things are. So we've got, we as an organismal biologist, my interests lie down here. And I'd say it's actually fairly late in my career that I was challenged to recognize that things like, and it played into ecology programs. So really like community ecology theory, that's where it's at, not natural resources. And so it's only fairly recently that I started to appreciate things like stream flow, sediments, these kinds of things might be important to driving what's going on in my system. So I'm sort of come late to the field, but But understanding, these complex systems requires a very interdisciplinary perspective, which is challenging but exciting. But it's also necessary, it's needed, this is, we could all find a, statistic like this to pull, but this one, we're almost, I feel like sometimes the ecologists and conservation biologists are competing. It's like, no, my system is worse off than your system. But this is like, my system is worse than your system slide. Because we're like, we are 84% of biodiversity loss, and we're laughing. But it's horrible, right? And all of our ecosystems are threatened, but freshwater systems are among the most threatened ecosystems globally. And so, broadly, what I'm going to talk today about is kind of some case studies where we're exploring like what shapes biodiversity in freshwater systems. These freshwater systems that we're interested in, and trying to understand why you have certain species here and not over here. This is sort of the classic fundamental questions of ecology, and sort of playing that out in this sort of riverine and adjacent floodplain rock pool context, to think about what are the drivers of biodiversity in this context. And there are also things that I think will be obvious that make this a really cool system for exploring questions of sort of classic ecology, where we might be thinking originally ecology, like we've got a habitat patch, and we're interested in what's in that. But we're now appreciating that, in order to understand what's happening in a particular patch, landscape context matters, connectivity also matters. And so this is a really cool system for classic island biogeography type ecology, because each of these little riverine rock pools is a patch of aquatic habitat, which becomes isolated from the river, but then reconnected with the river as the main channel sort of fills and retracts throughout the season. When we think about, what are the important drivers of why do we find certain species in a certain place, what are the drivers of patterns of biodiversity? We can think about broad categories of things like, abiotic gradients. I've talked to some of the grad students who are doing some really interesting thermal ecology, for example. Yeah, there we are. So differences in physiological tolerances in thermal biology, they can shape whether certain species can be in a rock pool or not, maybe, hypothesis. But then we might also think, because ecologists have thought about it for a long time, to the point of excluding all other things, there are things like competition, for example. It might be really important in shaping. These species are outcompeting each other, and that's what's really driving patterns in biodiversity. Ecology in the 70s and 80s was largely fixated on species interactions. And we have hopefully broadened our perspective, but that still remains an important component of thinking about biodiversity in a particular system. Our predator-prey interactions, our competition, these kinds of biotic interactions important. And then also, we are appreciating, as I kind of alluded to in that sort of, flyaway slide, that we're recognizing that patches are not really isolated patches. In fact, the whole idea that a pond is some sort of a boundary, that there's a pond ecosystem that is somehow independent from the terrestrial ecosystem around it, is a complete fallacy anyway. But we have people who are ecologists and riparian ecologists, and sometimes they aren't talking to each other. And yet we know things, from others' research that, forest feeds the stream, and reciprocal subsidies may be also important, where the stream is feeding the forest, and there's this interplay back and forth. And similarly, when we have patches of habitat, we're interested in dispersal and connectivity. Is a rock pool that's really isolated somehow very different than a rock pool that's in a cluster? This is a great system for thinking about those kinds of things. And then further, in the context of the current climate, we're thinking about aquatic systems. We're also wanting to ask questions like, well, how well alterations in flow, whether they're reduced flows or more variable flows, how will they impact biodiversity in these systems? As the world warms, what are heat stressors in these systems? Are they important for shaping biodiversity? And because we think about rivers as sort of being dynamic and connecting riparian habitats and retracting and connecting and retracting, how is that altered connectivity changing biodiversity? So these are all these broad kind of themes that we're interested in the lab group around the rock pool research. So the roadmap for today is first to sort of characterize certain dynamics of the system, because I want you to be intimately familiar with it, because I want you to come and collaborate with us. And the more that you know about the system, the more it might I'm interested in thermal ecology. That'd be a really cool question to ask in those rock pools. So I'm going to tell you a little bit about what we know about the system so that you can start thinking great questions. But then I'll give a couple of student examples of research in the lab group. One that's kind of focused more around abiotic gradients and connectivity and how that's shaping abundances of particular organisms and community diversity. And then a second example where we're looking at abiotic interactions of gradients and how they interact with species interactions. To what extent are species interactions dependent upon an abiotic context? So mapping, so this is the characterizing the system portion of the talk. So, I'm a third culture kid. I grew up in the South Pacific. I always wanted to be a tropical biologist. I did my master's in Uganda, my dissertation in Tanzania. My first grant was in Panama, always tropical. And so it's really interesting to be so excited about a system that's literally like less than a mile from my campus. But this is downtown Richmond, really within the city limits. And so when we started to, I was sitting with my grad advisor and he was saying, give a talk, 2007 probably. And it's like, James, these rock pools are really fascinating. You should do something with them. Like, well, now I'm getting back to that, right? So the first thing that we wanted to do is like, well, what do we have here? What is this system? Let's map and characterize the spatial context. If you think about, the geomorphology of the system and how that might be related to the hydrology and how that might be related to the biology. Let's start with that sort of gross geomorphology. And I'm not a geomorphologist, but let's map. So we use, drones that were georeferenced, and we created a digital surface model of the system and an orthophotograph, so that it's kind of the distances are real distances, and you can kind of think of this as like a topographical map that instead of it being like 100-meter intervals, it's like a couple inches, right? So that gives us this kind of really interesting fine scale variation in topography. These rock pools, some rock pools are on top of rocks, and some are down by the river, even though like if you look at straight line distance to the river channel, they're not actually different, right? And so just looking at sort of linear horizontal distance to the river isn't maybe as informative as also taking this into account. And it also allows us to think about things like clustering of habitats, right? So are rock pools even like somebody like run through the system and like punch a rock pool in at every 5-meter grid, or are they clustered rock pools? How are they spacious? So you see that there is some significant clustering where we have spots where there are lots of rock pools, but then there are other isolated areas as well. So we have some rock pools that are clustered, meta communities are sort of all tightly compact, and then others that are more isolated. And you can look at that. You can see, well, this is the distance to your nearest neighbor, and so this one's very lonely, right out here. But most pools, they have friends. The microtopography is kind of complex, and so sometimes it's hard to find your rock pool, isn't it, Karen? So one of the things that we did was we made a tool using Esri Collector so that we could help navigate the system. And so that's not at this point maybe so important for my lab because we've been working there for a long time, but we like to collaborate with lots of other folks who might not be as familiar. And so this is an app on your phone, and if you're standing next to the pool, it will show you standing next to that pool. Because again, we're in downtown Richmond and we have great cell towers. It's not like we're in the tropical rainforest. So this helps us navigate the pools, and it also helps us know that when we collect data on this pool, it's really that pool and not some other pool, because obviously that becomes problematic. So I'll show you, we have, if you were in the field here, you would click on that dot and then it would pull up a data sheet and you would start entering the data. I'll show you that in a minute. But this helps us navigate and it helps us collaborate, right? So that we can have multiple people collecting data in the system. And I'll get to that. So, so that's sort of so we have a map of the system now. There are 750 rock pools in the system in an area that's about a little bit bigger than a football field. And they vary in their topography and their clustering. What about hydrology? So, this is, we were just talking about this at lunch. So this is 3 years, and this is the hydrograph from, Weston Gauge, which is just upstream of this site. And so you can see that, there is some kind of spring consistent, like you get some higher level events in the spring and it tends to be drier, but like, look at the autumn here. Like this is super dry autumn, super wet autumn, super dry autumn. So there's a lot of variability even from year to year in the system. And then even within a year, you can see lots of variation. And the caption's gone here, but this is when, I forget what the percentile is, this is maybe like 75% of the pools flood at this height, and 50, I can't remember, but these are the different, like, the pools flood, the fraction of the pools that flood at these different river heights. So at this dark line, most of the pools are flooded, right? So you can see, most of the pools, none are flooded, they all get flooded, they all get flooded. Then there are periods where, like here, some would get flooded, like here, some got flooded, but others did not. So there's variation in flooding frequency. And the nice thing is that this gauge goes back, 30 plus years. So if you want to connect to flooding history, you can do that quite easily. So you can create these flooding frequency understanding for each individual rock. So this is just we use drones. And so we go out and we fly the drones at different river levels, right? Because we really wanted to know, we wanted to build a flood model. We wanted to know exactly when that pool flooded, so that we could connect that to the gauge data and sort of think about the flooding history. We started using some, Army Corps software, HECRAS, to sort of map that out, but it's really built for much larger spatial scales than what we're working with here. And so in the end, we kind of brute forced it, where we just went out and we flew a drone mission at a bunch of different river heights. And so we just took different pictures, and that's what you're seeing here. They're just kind of layered on top of each other. And then manually went in and we said, okay, river's at this height. Is this pool flooded? No, yes. No, yes, no, yes, no, yes, no, yes. And you do that for all the pools for a bunch of different river heights. And then you can start to generate a data set like this. So this is a logistic regression, probability of being flooded, river height here in feet, sorry. And so each of these is a different drone mission. So we flew it at, the river was at this height, flew it at this height, flew it at this height. And so this is the number of pictures that we had, right? And then you can do logistic regression adjusted in order to estimate the inflection point. And that's this river height is when we're going to say this pool probably floods around 8.5 feet, right? And so we can estimate for every pool the height at the river which it probably floods at. And then that was one pool. So you do that 700 times for every single pool. And every single pool now has an estimated flood height with sort of confidence run. Each time we fly a drone mission, we can improve the confidence, with that. That then produces a map. So here's a photograph of the site. And in this case, you can see that we've now color-coded the flood height of each of the different pools. And so you can see, like, these lighter color, these are pools that flood at low flood heights. And these warmer ones are ones that takes a good proper flood in order to get to. But like these two pools here, are about 10 meters apart. And so linear distance to the river channel doesn't necessarily, they're actually not correlated. Flood height and river distance to the channel are not actually strongly correlated. I mean, like 0.3, something like that. And then that then gives us the ability to connect to time. So here we have an example of April to 1 year. And this is what the river's doing. River's low, it floods, had a flood event. This is the river height. And then you can see over here, when the pools turn blue, they got flooded. So you can think about this dynamic nature of these pools in 1 year. Like they're winking, they're connecting, they're disconnecting, they're connecting, they're disconnecting. And that's all sort of, we can look at that. This is just 1 year, but you could look back, as long as we have river gauge data. And then we can think about this hydrologic variation, and we can look at it in a couple of different ways. We can think about, flood events per se. So this is how often was a pool, isolated and then reconnected. And that could be a disturbance event for some kinds of organisms, but it could be a connectivity event, a colonization event for other kinds of organisms, depending on what we're talking about. And so, you have pools that actually don't have very many flood events because they're actually always underwater. So they're never separated from the river. Then you have ones that are here so high that they almost never get flooded. So they also never get flooded, but for the opposite reason. And then you have these pools in the middle here where a lot of dynamics, they're getting connected and disconnected to the river a lot. So that's one way you can think about, the hydrologic variation, but you can also think about variation in the flood height. So these pools, they spend very little time in a year underwater, and these pools spend a lot of time under the river. And that varies from 1 year to the next. So you can really think about, well, what is this kind of connectivity? What is flooding? And it's actually, there are many ways you can look at flooding, and it might mean different things for different kinds of organisms. So that's sort of, now we've got the map, and now we've got the flood model. And then the last sort of, axis of environmental variation I want to present is the thermal environment. Where, as we think about, warming, the warming planet, we're thinking like, what are good systems where we can, in the field where we can start to think about how variation in temperature might be shaping ecologies or organismal biologies. And so, one of our challenges is security. This is a public, James River Park system. And so we have fancy, signs that measure DO and lots of stuff, but we can't really leave them out in these particular pools 24/7. So this is a data set from basically the grad student goes out at crack of dawn and puts a bunch of probes out and then harvests them back, before the end of the day, and then does it again and does it again. So this is really kind of like it's not a 24-hour cycle. It's really just kind of like morning to late afternoon. So they're capturing most of the variation. But you can see there's some really interesting. So this is basically on one afternoon. You can see a 20-degree variation in the max temp of a particular pool. And there's less variation in the min temp of those pools over that sort of daytime interval. And you have some pools that are really quite stable in their temperature. And if you think about the sort of pool morphology, that makes sense. You can imagine kind of like a narrow sort of tubular rock pool with kind of steep sides and shaded and goes deep down into the rock. It's pretty thermally stable, right? On the other hand, you might have a shallow rock pool. It's got a lot of surface area to the sun. It's not very deep. So that's going to, be more volatile in the temperature. So we've got interesting variation here in not only sort of on a given day, the maximum temperature that these pools are experiencing, but also like the variation. And we can also map that. So we didn't put hobos in all 700 plus 50 plus pools. So what we did is we essentially did that for about 200 of the pools, rotated that around, and then we developed a mathematical model to extrapolate to the other. But so now we have, call it not an absolute temperature model, but a relative temperature model for these rock pools, in the summertime are relatively hot ones, and these ones are relatively cool ones. So, so then so now we've got some of the important environmental gradients, hydrology, geology, the mapping, we've got thermal gradient, but ultimately we're out there to try to understand patterns in biodiversity. And so how do we sample that? So we use a sort of standardized protocol for two different kinds of data. We do dynamic occupancy sampling for presence absence, which I won't have time to talk about today. But for our sort of community data, we do sort of a series of standardized sweeps of the habitats. We call it 9 sweeps, because we do 9 sweeps for each rock pool. But we also want to try to standardize that across our collaborators. And so one of the tools that we have made is a standardized training video.
So here we are on the south side of Bell Island at the Dry Rocks area where there are hundreds of rock pools. And we're here today to learn how to sample the biodiversity of these rock pools. It actually doesn't take that much. It's not rocket science, it's rock pool science.
It's rock science, right? So anyway, we got rid of high school. So this is our, we make a training video. So you watch the training video, you learn how to identify the critters, then you can collect data with us. That's how it works. And to facilitate that, as I said, we use the collector app to help with the navigation portion of it. And then if you're in the field, you click on your touch screen, boom, and then it pulls up our data, which has Voshell's images from the common critters that they're likely to encounter. They enter the data on the phone and it goes up to the cloud. Now, this is pretty taxonomically pretty coarse, right? So that's one of the limitations of our data set here. It varies. I mean, some things we are getting to species because there's only one of them in the pools, but some of the other things are more like family. So the workflow is, you open up your collector app, you click on the data sheet, you collect your data, that goes up to the cloud, and then we can pull that down, or any of our collaborators can as well. And so with these tools, it has facilitated us collaborating with other organizations. So we're in downtown Richmond. Richmond Public Schools are a challenged school system, under-resourced school system. And so we've been very interested in sort of getting that near peer students college students interacting with high school students and doing science together. One of the ways that we did that was in collaboration with U of R. We ran essentially, a rock pool summer camp. This was last year of the MSI program, and they were calling for advisors, and they didn't get anyone except us. And so they said, "Would you like to take them all?" And we're like, "Okay, sure. We'll just do rock pool for everybody." And so we did a rock pool summer camp. And they all worked out in the field. They all learned basic macroinvertebrate identification. They learned some basic mapping because we're doing the mapping stuff, right? They're out here collecting in the field. And then we had, as a presentation at the end where they shared with their families and the community the science that they had done. And that was a really positive experience. Oh, sorry. So that led to another collaboration between VCU and Open High School. This was very rewarding. We were Emily Betts here, who was the high school teacher. She developed a class called Science in the Field, and we did rock pool, community ecology, case study rock pools. And we ran the university course and the high school course concurrently. And so it worked out that that undergraduates were just a couple weeks ahead because we started earlier. And so they learned to do the insect identification. And then we brought the high school class into our lab, and they learned how to do the insect identification and so forth. And the students had to do presentations, but instead of presenting only to each other, we went to the high school and they presented to the classroom and so forth. We had a number of students go on to do high school science fair projects out of this. Two that went to sort of the state science fair project, one of whom is now in my lab. Oh, and here's us doing, so here we've got, mentoring and identification, college students presenting in the high school classroom. We did actually a mesocosm experiment in their classroom. So basically this is a mesocosm. You might think it looks like a 5-gallon bucket, but it's really a rock pool. Where we tested to see whether two different snails differed in their vulnerability to crayfish predation. We also collaborate with U of R, going out to the rock pools and using some of the open source resources that we've produced with them is kind of part of the Standard Ecology Labs lineup. And now we've got a whole new dimension, barcoding, which we never ever imagined. And so that's super exciting as well. So this is, for those of you who haven't seen in field work, I'm going to take a moment to embarrass it. So what do we see in rock pools? We see all kinds of stuff, we see, once we got a snapping turtle, but that's not typical, right? But we do see, a variety of riverine introduced organisms. They could be fishes or they could be amphipods. They're things that can really only be dispersed into the rock pools if the river connects to them. And then we have a whole suite of other things. As a pond biologist, I like to call them pond stuff. But, there are things like, dragonflies and mosquitoes, and these are organisms that have complex life cycles that are aerial colonizers. And so they actually can't colonize the pools when they're underwater. They have to wait till they become pond-like. And then there's like this nice little habitat and they can colonize. So you have these different sort of regional species pools, if you will, like stuff that can colonize from the air, stuff that can colonize from the river, and how are they interplaying to create the biodiversity of these sort of unique ecotonal kind of edge habitats. And at this point, and this is not like for science data, but we also have a BioBlitz that we do down in the rock pools. And so, we have 111 critters that we've BioBlitz out of there. But the actual science data set is this. So this is based on, we've seen a total of 36 taxa in the rock pools over the years. And that's based on sampling 500 rock pools plus, multiple times. So essentially we have more than 1,500 community data sets, right? For those of you who do community ecology, you'll appreciate that it's not very often that you have 500 communities sampled with that level of resolution temporally. And we have that over now 9 years worth. And this is what the distribution, so total species richness in pools, we see typically there's not a ton, about 5 or 4 species on average, but some pools have as many as 15, and some don't have anybody. So that's kind of the stage. Right? That's what we know. We know a little bit about the system, and I'd love if this kind of connected with any of the kinds of stuff that you're doing, in your lab group, I would love to brainstorm fun collaborations with that. But now let me get into just kind of two examples of some of the research that we're doing, given that context. So grad student Ryland was a master's student and now is a PhD student. He's really started out as being interested into how do species traits, kind of coming back to this broad characteristic that I alluded to in terms of complex life cycles and how they're colonizing pools versus sort of more aquatic or marine. How does that shape difference in responses to flooding? Because you might think that fish might think of flooding as a different thing than a frog that's colonizing these things. And so when we, can we actually see that signature in the data? Does that hypothesis bear out? So, here I'll give you, a gilled snail and a dragonfly larvae. So where would you predict to find these pools, to find these organisms in the rock pools? So the audience participation. So where are we going to find gilled snails? What would you contrast these to? And if you've been to rock pools, you're disqualified.
Because of how frequently they're flooded.
Okay, so which one would be where?
Snails are almost like exclusively aquatic. They're not going to be on the edges where there's water.
Thank you. Yeah, for example. So this is an obligate aquatic organism looking for hypotheses like highly connected rock pools, maybe down next to the river. This is where we're going to find these kinds of snails. Dragonflies, perhaps, maybe the opposite. One could hypothesize, right? And there's a whole suite of organisms that kind of match these broad life history categories. So this is what he was interested in looking at. Because these, only able to move through patches when they're connected. These can colonize airily. They can have some interesting seasonality too. The most common ones are actually migratory in our system, and so forth. So we can make some predictions based on this, that these obligate aquatic organisms like the snail, we would find them if they are at low flood heights, we might find more of them. If they're at high flood heights, up high, they won't get connected very often. So we might expect their abundance to be correlated in this way. The opposite direction here, maybe distance to the river. Again, close to the river, you see lots of snails. Far from the river, maybe the opposite here. Time isolated from the river. So we actually know, we actually have the interflood interval for every pool. So we can say this pool on this day, it was last flooded 6 days ago. And so we can think about time isolated from the river. And again, very isolated ones, because these might, they might start out being okay in a rock pool, but as it warms and the oxygen goes, down, they might sort of start to drop out of the picture, right? And then it becomes like, well, how quickly is it reconnected to sort of rescue them? Not necessarily by colonization, but maybe even just by an influx of cold, oxygenated water. So we made these predictions, and here's what we found. We found evidence to support, we see a sort of species abundant, like there are more higher densities of snails in larger pools, but we didn't really see any relationship between time isolated, but we did see that snails are more abundant in lower pools that get flooded more frequently and that are closer to the river. So as one might expect. With the dragonflies, we had sort of the opposite predictions. And we saw some support for, pools that have been isolated from the river and have been sort of open for colonization longer, tended to have a higher abundance of dragonflies. But flood height was not important, nor was distance from the river. So we do see some evidence about these organisms with these broad life history traits kind of responding in ways that make sense to us, but not all predictors supporting in the direction that we found. So if you start to think about that, then we started to kind of think about this cartoon of like understanding the biodiversity in rock pools, that they might start out as essentially being riverine communities. Like the river comes up, stuff that's in this river for the most part. The river drops down, it's a riverine system that's being isolated. But then as time goes on, maybe some of those species start to drop out because of physiological intolerances, and maybe some other stuff starts to colonize. So they're kind of like these riverine habitats that are walking toward becoming ponds, and whether they do or not, kind of get reset back, depending on whether they get connected to the river or not. Again, just kind of a cartoon way to think about it. But if we kind of think about these two organisms as exemplars, do we find that pattern bears out? So you can kind of make the same predictions, not looking at just two taxa, but kind of all of the organisms that we think will be dispersing through the river and all of those that can move over land. And this is sort of a different way to look at those data. So here we have an effect size plot. So we've got like things over here. They have a negative effect on biodiversity overall. These have a positive effect on biodiversity overall. And we're going to look at the biodiversity of pond things, river things, and all biodiversity together. And what we see is that, when we're looking at river stuff, the longer they're isolated, the fewer species from the river we find. That makes sense. The larger pools have more riverine biodiversity, so species area relationship. Oops. Interestingly, this is counter to our expectation. We would have thought that these would have maybe been more cold tolerant species, but we actually see that more riverine species in warmer pools. We also see sort of a seasonal drop off. We have more riverine stuff early in the season. Spring's not shown here because that's the sort of the other variable. But so they're dropping off as we get into summer and fall. We're having negative effects on their biodiversity. We see the seasonal effect of biodiversity. And then there's also this positive canopy effect. And if we look at the pond stuff, we see mostly the main signature at the richness level is that we see warmer pools have more ponds stuff. The other variables and more canopy also facilitates, which might be related to sort of nutrient inputs. Perhaps we haven't explored that. Less of a seasonal effect and really no effect on overall of the flood hydro period components. And so then when we look at the overall diversity, we see the overall biodiversity is sort of lower in pools that are flooded frequently, higher in larger pools, sort of species area relationship, higher in warmer pools, tends to decrease a little bit over the season and is positively affected by canopy cover. One of the first things, that's sort of a correlative. So we've gone out into the field and we've looked at these patterns. We haven't done an experiment as yet. So one of the things that's really cool about the rock pool system is it is actually somewhat amenable to manipulation, even in the field, in addition to being able to use mesocosms as sort of a proxy. So we are in the process now of essentially repeating the classic Simberloff and Wilson study. They essentially use methyl bromide to defaunate the invertebrate faunas from mangrove islands that were different distances from the mainland in Florida. This is one of the classic empirical studies of island biogeography. They looked at colonization afterward. And this was sort of the study that really supported the ideas of island biogeography. So you can kind of do the same thing in rock pools and a little bit less invasively. I mean, we basically can pump out the water, remove the sediment, transfer it to the river. We don't use pesticides. Scrape down and scour the sides, and then get an empty rock pool, and then sort of do those at different distances from the river. Sort of repeat the sort of classic island biogeography study. And we've done that for 20 pools that are defaunated, cleaned out, and 20 that are control pools, and we've been following their history. All I can say at this point is that we see a fairly rapid, the defaunation did reduce the diversity quickly, but we actually see a pretty quick rebound. And we need to we're in the process now of parsing that out into pools that are close and pools that are far from the river or their flooding histories. And then for the last example, I'd like to sort of get to this idea of abiotic species interactions. A lot of the work in my lab group focuses on predator-prey interactions. In particular, this study is sort of like, what's the role of predators as the world warms? And it kind of actually starts out with a conceptual model that is thinking about the effects of predators on prey as temperature changes. It's often been sort of linked to the sort of thermal performance curves of the predators. We focus on especially ectotherms. As it warms, you're going to ramp up the metabolism of the ectotherms potentially up to some threshold. So warming may, depending on the sensitivity, this is not a very sensitive predator. This would be a very sensitive predator. So warming here would really ramp that predator up. Warming here, not so much. And so it's these sort of differences in the predator's sensitivity to warming that will really drive the strength of that warming effect on top-down predator-prey interactions. But what's been less appreciated, but is obviously a truism, is that the prey are also being warmed. And so in these systems where we have many organisms with complex life cycles, It's also important to consider how warming may alter, for example, their developmental period. So if you have, for example, development time in the pond, this is essentially, or the pool, this is the window where they're vulnerable to that pond predator. When they emerge, they're not vulnerable to it anymore. And so if you speed up development, they're going to spend less time in the pond, therefore less time exposed to that predator. So you can also think about, for example, how the prey traits are sensitive to the thermal changes. And so if they're not very sensitive, it may have little effect on the time that they're exposed to a predator, but if they're very sensitive, it could have a big impact. And so how do these two things interact? We argue that really to know how warming is going to affect a predator's effect on its prey, you really have to have both of these pieces, and either one could be dominant. And so this is the conceptual model. And we wanted to, and I guess this is, so these two things could combine such that increasing temperatures, actually leads to more prey surviving. So, i.e., it reduces the effective predators, or warming could lead to fewer prey surviving. And so that would be, and warming enhances predation. Either is possible depending on the relative sensitivities of predator foraging and prey development to changes in temperature. So you got to know both of them. So our sort of focal organisms here are the predators of globe skimmer dragonfly, the most common dragonfly in the system. This is the business end of it in the rock pool. And then we're also focusing on the Native American rock pool mosquito, Aedes aegypti. My favorite mosquito, this mosquito does not need a blood meal in order to make eggs. And so it's a great lab critter. You can just kind of have your colony of, essentially, and they're related to obviously important disease vectors, but they're not important disease vectors themselves. So it's a great system to work with in that regard. And they're also super common in rock pools. They are in fact the North American rock pool mosquito. So first we look at the thermal tolerance of the predators. So again, this is the natural range of temperature, which is what we're looking at here. And we're basically asking how sensitive is this predator? And so this is a cold temperature, this is a warm temperature. So you can see that as we increase the temperature, the number of prey killed is increasing. So more killed here than here. So this difference here is a measure of the temperature sensitivity of this predator to changes in warming over the range that's seen in the rock pools broadly. What about the prey? Here we're looking at the prey, and we see this is time for them to metamorphose and fly away and not be vulnerable to this dragonfly predator anymore. And we see that in cold pools, it takes them about 2 weeks to metamorphose. In warm pools, it takes them less than a week. There's a huge effect on larval development time, so they spend a lot less time exposed to the predator. So when you combine these things, and there's also, but when you get to really high temperatures, they die. So there's that upper limit. So these experiments were done on sort of a 24-hour period. So we use a little bit of mathematical modeling to kind of crunch that through to say, like, under these different scenarios, how many mosquitoes emerge given that there's a predator in the system. And this is what the model shows, is that we see that at low temps, predators have a big effect. This is survival of prey, not low survival of prey at low temperatures. Predators big effect when it's cold. As it gets warmer, we actually see that predators have a less of an effect. So temperature actually is reducing the effect of predators, in this case, the top-down control of a natural predator on a mosquito, because it's basically having a bigger effect on ramping up development than it is on ramping up predation. And these two things, they're asymmetrical. And so we then can sort of crunch that through. But this is still like, these are done in incubators, and then we've got some math. This is all sort of not in the real world. This is really a model prediction that's been based on experiments in the lab. Can we see any evidence that there's weaker predation in cold, so weaker predation in warm pools, or more predation in hot pools when we go out into the field? And we have abundance data on mosquitoes and dragonflies as well. And so what we're looking for here is essentially kind of an interaction between the effective temperature and the effective and the relationship between the dragonfly predator and the prey. And we do see some evidence that's consistent with this, which is kind of shocking given how noisy these systems are. And there are other alternatives for this. But what we see here is the difference here. This is like the effective temperature, in the absence of a predator, which interestingly is positive, which is different than what we see in the lab. But let's not talk about that part. Let's focus on this part, which matches the predictions, where we see that basically how big this arrow is that's the effect of the predator. No predator, predator. Cold temperature, we see a big arrow. There's a big difference from here to here. Warm temperatures, small difference. So big effective predators at cold temperature. Again, I'm being a little loose here by calling it an effect of temperature. It's really, this is the relationship between the abundances of the predator and the prey across a gradient of temperature. But it is consistent with the idea that they're having a bigger effect at cold temperatures and a weaker effect at negative temperatures, which I think is super cool because we've gone from a conceptual model to laboratory experiments to a mathematical model, which extrapolates that to metamorphosis, to looking for patterns that are consistent over hundreds of pools in the field. And we see some evidence that that may be the case. So to wrap it up, importance depends on traits. This is the first part. So really thinking about biodiversity in the rock pools, we see that some interesting differences between pond and riverine organisms and how they're segregating across the system. And it does seem to be related to differences in connectivity, and warming was also important. So those seem to be important community drivers. And then also some really fascinating interactions between the abiotic temperature gradient and how that's predicted to impact the relationship in kind of a counterintuitive way. Warming, because most people focus on the warming effect on the predator, which would predict a bigger impact of predation. When we focus on the prey, we see the opposite pattern, and we not only sort of predict that in the lab, but we see evidence for that in the rock pools as well. And as a final slide plug, I will say this is a study I didn't have a chance to talk to you about. We don't only do rock pools in Richmond. Rock pools are a pathway to world domination. And so keep an eye out, because this is a class that's open to anyone. It's a field course that we teach at a Highlands Biological Field Station, which is not a sad place to spend time in the summer, where we actually have a population of meta-communities along an elevational gradient from about 1,500 all the way down to the Georgia Piedmont. And so imagine what I've shown you in the Richmond rock pools, but now at a gradient with 7 different sites in the Chattooga River, wild and scenic river, not ugly, and sample rock pools as you sort of travel down. And spending time on rivers is not at all anything I'm interested in, by the way. And with that, I will conclude and thank you and invite you to come collaborate with us. Didn't leave a lot of time, I apologize. Got one question there, though, if I can grab one.
So when you were discussing the effects of rock pools on community diversity, you had mentioned that there was a connection between rock pool size, temperature, and dissolved oxygen, and then also flora and fauna. Did anybody look at possibly doing any kind of covariate analysis on all of those? Because as you have a larger body of water, you anticipate a decreased rate of loss of dissolved oxygen and a decreased rate of increase in temperature. You have to
Great questions. So for the community, so we have not done that in part because we don't have the data. We don't have dissolved oxygen at the scale at which I'm talking about. We have done, I had a master's student who deployed sort of 24-hour sondes, and we sort of did diurnal oxygen profiles to estimate essentially ecosystem metabolism. Like each of these is an ecosystem. Like if you're wanting to do ecosystem stuff, like you've got hundreds of them. Again, the ecosystems that most people don't care about, but let's not get free bothered by that. But so we have looked at that, and so that work did give us one of the things that we found is there's some really interesting variation in sort of rock pool ecosystem level metabolism. But based on that student's work, there weren't any strong predictors of what was driving that variation. So when we look at these kinds of things, they just don't pull out. Now, part of the issue is that this is a super noisy system. And when you think about, ecology and community ecology, we talk about a lot about equilibrium systems and sort of stable ecology. This is not what's happening. I mean, these are very dynamic systems, and the idea to even sort of like impose sort of classic community ecology equilibrium thinking upon the system is challenging. But so what I got to talk about today is like thinking about how much of these patterns is actually explained by these predictors. And actually, like if you look at the pattern of, say, community biodiversity, it's explaining like 30%, right? So the R squared is not an R squared, it's a different metric, but it's actually very low. And so, why is that? Part of it could be because we didn't include the right things, but also part of it is because it's a highly stochastic system. So, are communities governed by rules, or are communities just chance? And the answer is yes. I would say, and trying to parse out the relative contributions of deterministic, predictable processes versus stochastic, random processes in the system is actually a really interesting question. And another question that I think the system is actually quite amenable to, but I didn't talk about it.
I might have missed this in the beginning when we were talking about the sort of structural assessment of the different pools, but like turbulence is obviously a huge one here in a riverine system.
Yeah.
So how does that affect connectivity? Because I imagine, turbulence goes up, connectivity is going to go up between pools, or isolation is going to drop.
Yeah. They actually, you really kind of highlight. I came in thinking about like flooding as a disturbance. I'm a pond guy. Like floodplains hate flooding. They want to get out. And so, when you think about the morphology of the pools, you can imagine like how is turbulence differ between a shallow pool versus a really deep tubular pool, and how the turbulence lens, how deep does it extend down into a narrow tubular pool? Now there is some publication on that, not by us, that we use to sort of inform to motivate, I guess I would say, the prediction to say, for example, depth would be important, because that would be kind of like, is there something inherently about being a shallow pool relative to your surface area? That's kind of a real rough, pool morphology measure, like surface area to depth, right? That stuff doesn't pull out. When we put it in, volume is more important, so like just big volume, which is obviously correlated with the things that you talked about. But if you look at depth per se, it doesn't seem to be a strong predictor of diversity. But you might say, for example, like depth, and because there are actually some of these pools are big enough you could have a thermal stratification. And these temperatures, I should say, are basically measured at this point by putting a temperature logger at the bottom of the pool. So it's probably the coolest the pool gets. And so
But things that live in rock pools are usually like extremely thermally tolerant. They don't have as like restricted thermal neutral zones, things like that. So
yeah.
just curious about that. Okay.
So, I really enjoyed the talk. I mean, it was impressive that you could involve so many people at different levels, but and also hearing your final comments about communities having structure versus being stochastic reminded me of the Clemens versus Gleason story when I learned ecology 40 years ago. But the question I have is, what about rainfall in terms of being something that could temporarily reunite pools and bringing things from the opposite direction, not from the river into the pools, but the other way?
Now, that's a great question. And we actually have some so we've done some experiments. One of the students is looking at hydrilla is endemic into the system. And so we're looking at, he's done a sort of dynamic occupancy modeling using 3 years of data for all of the pools, where we're basically predicting extinction and colonization dynamics of hydrilla across the system. As part of that, he's done an experiment where he's inoculated hydrilla. And one of the things that we've seen anecdotally is there are cases where exactly what you described happens. If you get a scenario where the pools are relatively full and you have a rainfall event, the pools can overflow. And if you have floating organisms, snails will move along water trails. That definitely is, that's happening for sure in the system. And it's something that we haven't captured as yet. Even with our fine scale topography, it would need to be finer stuff.
Thank you so much.
Thank you.
Sold the system really well.